Good afternoon. My name is Allison Clune with the U.S. EPA's Indoor Environments Division. I'd like to welcome you to the IAQ Science webinar series hosted by the U.S. EPA. Today's webinar, The Influence of Clothing and Other Textiles on Your Exposure to Indoor Air Pollutants, will be presented by Dr. Glenn Morrison. Dr. Morrison is a professor of environmental science and engineering at the University of North Carolina, and he's been studying indoor and outdoor air pollution and human exposure for 30 years. He received his PhD from Berkeley, and his research has primarily been related to indoor physics and chemistry. He has a particular expertise in interfacial chemistry, having also worked as a chemical engineer on heterogeneous catalysis. This research has included ozone surface chemistry, acid-base chemistry, and its role in absorption to indoor surfaces, methamphetamine contamination in buildings, aerosol SVOC modeling, and field measurements of reactive oxygen species in homes. In recent years, he's focused on the role of clothing on indoor chemistry and occupant exposure. Dr. Morrison has organized numerous workshops to bring outdoor and indoor chemists together to discuss indoor chemistry and indoor air 2011 in Austin, Texas, as Vice President and Technical Chair of the conference. From 2014 to 2016, he was the President of the International Society of Indoor Air Quality and Climate, or ISIAC. Thank you so much, Glenn, for being here with us today. Please note, this webinar is being recorded and will be made available on the EPA website. This presentation reflects the opinions of the speaker and not necessarily those of the U.S. EPA. Before we start, I'd like to ask Christina, who will be running the webinar today, to go over a few housekeeping items.
Certainly. Thank you, Allison. So today at the end of the webinar, we will be having a question and answer portion. Please submit all questions using the chat function, which is part of the webinar program. All questions will be submitted to our speaker at the end for answering. Thank you for joining us today.
Thanks, Christina. And with that, I'll hand it over to you, Glenn.
Great. Thank you, Allison. Thank you, Christina. All right, so let's get started. So today I'll be talking about clothing and exposure to indoor air pollutants as influenced by clothing and some other textiles. I wanted to make a note before I get started about what I won't be talking about, and because this is actually a really, really big subject. There's a lot of interesting phenomena that can occur when you're starting to look at textiles and human health. And so this is a fairly focused presentation. So I'll just be talking about the kind of pollutants that you find indoors and how clothing and textiles influence your exposure. I won't be talking about, in particular, the kind of pollutants that actually are part of clothing when you purchase them, that come from manufacturing. Some of those might come into play anyway, but that's not the focus. I also won't be talking about, in particular, particles and secondary exposure to particulate matter or biological materials that come from clothing. So let me get started. So when we talk about exposure and environmental exposure, we need a couple of things to have in place. We need to be in contact with an environmental compartment. And what we mean is basically, if you're touching the air, you're in contact with that compartment. Or if you're touching a couch, you're in contact. But we also need another thing we need the thermodynamic activity of a chemical to be higher in that compartment than in you in order for it to move from that compartment to you. So we call this thermodynamic activity either the chemical activity or the fugacity, but the name isn't so important as thinking of it kind of in terms of the intensity of what's there is higher than in you, and so there's this tendency for those pollutants to move from that compartment to you. This is a bit like temperature. If an object is hot and another object is cold and they're in the same environment, there's a tendency for heat to transfer from one object to the other. Same kind of idea. Another way to look at this is, and one of my favorite ways, to think of these as reservoirs. You can think of kind of containers connected by pipes. So there are big containers and small containers, but the height of the containers is related to this idea of chemical activity. So if we have, let's say, a couch that has a lot of a particular chemical in it, let's say a flame retardant, it might have a high level, a high activity, but also it might have a high capacity. It's a fairly large reservoir. The air may have a fairly high intensity of a compound, but the total amount that's there is small because it can't hold on to very much. And then there's you, and you may be somewhere in between. It depends on the chemical, but for many chemicals that we are concerned about as pollutants, the activity in you is generally lower than the environment because your body is taking it in and oftentimes metabolizing those chemicals to something else. So the chemical activity is what drives the sort of direction of the exposure. So if the height of these reservoirs is our chemical activity, then you have a high chemical activity, let's say in the couch, low chemical activity in you, but because of that gradient, that difference, there's a flow of the chemical from that couch to you through the air in this case, or perhaps you could be in direct contact. Now, how would you reduce that activity? Or how would you reduce this exposure or this net chemical uptake? Well, one way to do that is to simply get rid of the chemical to begin with. Sometimes that's not so easy, but maybe we can lower the activity in each of these compartments that surrounds us in a way that we have less going into us. So that might be lowering the chemical activity of the source, in other words, removing that or reducing the amount we use in the product's amount of a particular chemical that we use in our consumer products. Or perhaps we have something that draws the concentration down in the air around us. Maybe there's a air cleaner, or you open a window, that kind of thing. Ultimately, this reduces our uptake. Now, I wanted to put it in these terms because I'm going to use this analogy every so often throughout this presentation to give you guys an idea of what's going on when clothing is involved. So let's talk about dermal uptake from the indoor environment before we talk about clothing. So chemicals in the air can move from the air to us, and some of them can move through our skin. Now, if we take a section of our skin and start looking at it more carefully, the skin actually is kind of a barrier to a lot of chemicals. But there's more to it than just our skin. So outside our skin is something called a convective boundary layer. There's this region of slow-moving air next to your skin, and this is really over any indoor surface. There's this, whereas the air around you is kind of mixing at a low velocity around you, when you get closer and closer to a surface, the velocity gets lower and lower, and eventually zero when you actually get in contact with that surface. So this is an area that slows down the transport, the movement of chemicals from air to skin. In the skin, there's a layer right at the top of mostly dead skin cells called the stratum corneum. It's a really interesting region of the skin. I won't go into the details, but basically it acts as a natural barrier, a human barrier to chemicals. It actually is really good at holding in the water in your skin, and so that helps keep your skin hydrated. But it also happens to be a good barrier for many chemicals as well. So even though we have these barriers in place, some chemicals can get through both those barriers into the skin, and the rate at which they can move in really depends on a lot of parameters, but a lot of it has to do with the chemical structure, the structure of that chemical. So if we go back to our analogy of these reservoirs, let's say the air has a fairly high chemical activity of some chemical in the air, and we're at some lower chemical activity. In between are these two resistances, this resistance associated with the convective boundary layer, this sluggish air over your skin, and the stratum corneum at the top layer of your skin. If we take a look at it and break those two down into two separate resistances, you can think of them as two different pipes with different diameters between these two reservoirs. For a chemical like hexane, that's just shown at the top right, it turns out our skin is a really good barrier to hexane. It doesn't really let much through. It does let a little through, but it's like having a much smaller pipe between the reservoirs. If we look at a different chemical like this one here, this is a plasticizer, and it's has been historically used in a lot of products, although it's been being phased out. This dibutyl phthalate actually moves quite readily through the skin. And you can think of that as basically being a pipe for the skin in this sense, has a bigger diameter, and so there's less resistance from air to you. So a few years ago, Bill Nazaroff at Berkeley and Charlie Weschler at Rutgers, they were really interested in the idea that there were compounds out there that might be able to do this. In fact, they were the first to really hypothesize that many of these, what we call semi-volatile organic compounds and others, can move from indoor air through our skin in a way that's meaningful, actually in a large way. And so what they did was they developed this figure. I won't go into the details of this figure, but you can see here that there are a lot of chemicals with different molecular weights, and they have this other parameter here, the stratum corneum gas phase partitioning. This is a thermodynamic parameter that is related to how much is likely to be able to equilibrate with the top of your skin. They classified these by something called a permeability coefficient, where these over here have a very high permeability coefficient through the barriers over and in your skin, and those that have a low permeability coefficient. It turns out that for a large number of chemicals that we do see in the indoor environment, the permeability is high enough for this to be a really meaningful exposure pathway from air to through skin and into your body. So let's put this in context of breathing. So if we take a typical person, they inhale a rate of about 0.6 cubic meters per hour. Actually, I'm not quite sure what that is in cubic feet per minute, but anyway, it's about 0.6 roughly cubic yards per hour, I guess you could put it. So how does that compare to how much you take up through your skin? Well, we can take this permeability coefficient that Weschler and Nazaroff estimated from the chemical properties of these compounds. We can take that value and we can multiply it by the surface area of the skin of a person, which is about 2 square meters. When you do that, you get this value. This is how you would actually do this comparison. 10 cubic meters per hour equivalent uptake from air of these chemicals relative to much less by breathing. Now, this does not mean that air is passing through our skin into our blood, but what it does mean is that the chemicals are passing through our skin into our blood at a rate that's equivalent to 10 cubic meters per hour of breathing effectively. So it's a lot more, and that means that this is a pretty important exposure pathway for these chemicals, or potentially. Let's put it that way. When this was hypothesized, these experiments had not quite been done. There was enough information out there to say it was probably true, but the validation in human participants had not yet been done. But we have to be careful. This calculation here really is a very chemical-dependent calculation. Some chemicals, like I said, something like hexane has a very low permeability coefficient, and so the effective amount that goes into your body would be a much lower than 10 cubic meters per hour. So it depends on the chemical. So how do we test this hypothesis? Well, what you do is you get some volunteers. And this is what the folks at the Technical University of Denmark did a few years ago with Charlie Weschler. And they designed a set of experiments to actually test whether they could really see that these chemicals in indoor environments would actually go through the skin at very large rates, more than our intuition would say is possible. So this is what they did. They used 2 chemicals for this test. They used something called diethyl phthalate up there on the top left, this guy right here, and dibutyl phthalate, that one right there. And they put both of these into the air around these participants. And so this is basically a room that they elevated the concentration of these species. The way they did that was for basically there was these panels that were painted, and they had these chemicals in them so they would emit into the room. That method and the analysis of the air was actually done by a group in Germany from the Fraunhofer Institute, WKI Fraunhofer Institute. So essentially what they did was they had these participants, these volunteers, go into a chamber with an elevated concentration of these chemicals. But note that I have these little space people here they have this square over their heads. That means they're wearing a breathing hood. And the reason for the breathing hood is to provide them with clean air so that whatever is seen in this experiment in terms of absorption from the air comes from dermal uptake only through the skin. So they go into this chamber for 6 hours, and they're also not wearing very much in the way of clothing, just wearing shorts so that most of their skin is exposed. After 6 hours, they leave, they put on normal clothing, and they go about their day. A week later, they go back into this chamber, same conditions. Everything is the same except they're not wearing the breathing hoods. And this is so that you can see the difference between with and without. And so the difference between these two experiments tells you something about the inhalation rate of these chemicals. So just to reiterate, there were 6 subjects. They spent 6 hours in the chamber, once with the hood and once without this breathing hood, these two chemicals. And the other thing I didn't mention is that they had a restricted diet because some of these chemicals can actually come from food. And so the idea was just to reduce the possibility or reduce the background concentration that we get from other sources as much as possible. This is what it looked like. So you can see the breathing hoods there. You can see that the volunteers are wearing shorts, but most of their skin is exposed. There were a variety of decisions made about what should be and shouldn't be exposed, but this seemed like a good compromise. One thing I also didn't say was that they collected urine throughout the experiment from before, during, and after the experiment for about 54 hours, I believe. And the idea being that we can use the information from the urine to tell us how much is absorbed because these chemicals metabolize in the body to unique metabolites that are then excreted in the urine. From the urine, you can see it's at least gives you some idea of the rate at which these chemicals are absorbed. So this was the result from, at least a result from one individual from that experiment. And what you see, each one of those points on this figure represents a urine collection. On the y-axis is the amount of a metabolite excreted. This is MNBP, the monobutyl phthalate. It's relatively easy to see in a urine sample. And on the x-axis is the time axis. This is a cumulative amount, so what's happening here is that the curve represents the total amount. When you're done at the end of 54 hours or so, 54 hours I think, the last point represents the total that was absorbed. This also accounts for the background amount that you see in urine before the experiment, so that was actually subtracted out. So the orange result there is where the subjects are wearing a hood, and this is where the subjects are not wearing a hood. So this is inhalation and dermal uptake. This is only dermal uptake. And what you can see immediately is for this chemical, the amount that you see absorbed, and this is proportional to the amount absorbed, it's basically what mostly what you see here is what it was absorbed, is that the dermal uptake is as much as the inhalation. The difference between these two is related to inhalation. What's not shown here is that if the experiment itself, the actual exposure during the first 6 hours had been allowed to proceed for much longer, the dermal component of this would actually increase quite a bit. And I won't go into the details of why that is, but it has to. This was not what we call a steady state experiment. It was a short dynamic experiment, which only gives us a hint about how much dermal uptake would occur. So now we've confirmed that we can see this uptake through the skin from air. Now let's finally get to clothing. What's going on there? So we do know something about clothing already before we even went into these experiments. There was evidence that chemicals that had been applied intentionally to clothing, it can be absorbed through the skin. There's evidence for flame retardants in children's sleepwear, which is one of the reasons why flame retardants were removed from children's sleepwear. And also pesticides in uniforms. There are pesticides applied to Army uniforms or military uniforms to prevent mosquito-borne disease transmission, and you can see those pesticides in the urine and blood samples of those soldiers. So that's one thing. We also know something interesting in that clothing will absorb chemicals from air. And the first sort of hint we had of this, well, first hint that I had of it was when we were working with methamphetamine when I was working in Missouri at the University of Missouri. Many of you probably are familiar with the problems that many states have had with illegal meth labs. And these meth labs contaminate a building with a lot of chemicals, but one of those chemicals is methamphetamine itself, and over time it can accumulate in the building. Even if you clean a building, a small amount can still be released from building surfaces over time. And so we were interested in what would occur if somebody moves into that home and clothing is present. Would it accumulate in clothing in a way that was meaningful? And what we learned was that it did. You'd see a lot of methamphetamine, basically absorption into clothing. And since then, there's been quite a bit of work done here in our institution and in others, looking at the amount of these chemicals that can absorb into clothing. And it turns out that the capacity for clothing is quite large. This is a result from some experiments that Azina Eftekari, my student, did actually a couple of years ago now, in which she was studying how much these chemicals can basically absorb into clothing. And what you find out from these experiments, you don't have to look at the details there, but the concentration in clothing is about 1 million times greater than the concentration in air once it gets to equilibrium, depending on the chemical. The smaller the molecule, the less will absorb. The larger the molecule, basically, the more will absorb, roughly speaking. And so for many, many chemicals, we see this accumulation and a lot of accumulation. So once we know that the chemicals can move from indoor air to skin and through skin, we know that chemicals that are on clothing can move into skin, and we also know that indoor air, contaminated air, can contaminate clothing. This probably means that the chemicals that are in indoor air will eventually accumulate in clothing, and that will be a direct exposure route for those chemicals and us. So we had a hypothesis. We had two hypotheses, actually. We probably had quite a few more, but these are the ones I'll talk about. First is that when you have contaminants in indoor air, clothing can be protective, potentially. It can be protective because it has such a large capacity for these chemicals. It acts like a sponge and maybe prevents the contaminants from getting to you. So this, in essence, would reduce total dermal uptake if the clothing does not already have these chemicals in it. So using our analogy of reservoirs, it would look something like this, where you have a high chemical activity in the air of some chemical. The clothing is at some low activity. Let's say it's relatively clean, and therefore you would see a net chemical uptake from air to clothing. But because the clothing is on us, we would actually see maybe not so much uptake from clothing because the chemical activity is still low, and maybe even if you do it right, it's even lower than us. So that's how it would work. So for a second hypothesis, if the clothing eventually absorbs enough of the chemicals from air, it will then be a source and perhaps enhance the dermal uptake relative to bare skin. So that would be something like this, where the clothing basically has reached equilibrium. It's at the same chemical activity as air, and so there's no real net transfer from air to clothing, but there is net chemical uptake from the clothing because of this gradient that flows from the clothing to us. Analogy that I've used in the past I kind of like is this one, where if you have clean clothing, that's kind of like a tree that is dry, but you stand under it in a rainstorm and it keeps you dry initially, but over time, that rain saturates the tree and then the rain starts coming through and then you get wet. So, all right. So how did we test this? So this looks exactly like the experiment I showed you before. In fact, these experiments took place at the exact same time as the experiments I described earlier. What we did was, when the folks in Denmark planned their set of experiments to study how bare skin absorbs chemicals from air, we added in a little bit more to that and added in some clothing experiments. So in this one, imagine this one right here is actually is blue. So this one is actually wearing clean clothing in the first week of exposure, and then the same person the next week is wearing clothing that has been exposed to the air in this chamber that has the phthalates that I described earlier. In this case, the person who's wearing clean clothes or wearing the basically contaminated clothes, in a sense, the clothing that has interacted with this environment, in both cases, that person is wearing a breathing hood, because in this case, we only wanted to study the dermal side of this. We're only changing one thing, and that was the state of the clothing from this one to this one. For those who are wondering, this was actually cotton, 100% cotton clothing in this particular experiment. This is a picture of the participant here. And note the coverage, pretty well covered by the clothing. This is actually the clean clothes here. We didn't use gloves in this experiment. In retrospect, it probably would have been better to use gloves, but we were learning at that time. And we see, note here, this is the clothing for the next experiment, where this individual, acting as their own control, will wear these clothing the next week after they've accumulated these chemicals from their environment. So this is the result from that experiment. This orange line here, this is the bare skin result that I showed you earlier for an individual. So we can compare the clothing results to the bare skin results. This kind of grayish line, circles and dotted line down here represents the results for the clean clothes. The amount of this excreted, this metabolite excreted, is much, much, much lower if you're wearing clean clothes in that chamber. There's a slight rise here, and that was probably because that individual was not wearing gloves and it was absorbing the chemical through their hands. For the second week, when they were wearing the exposed clothing, the amount that's taken up by the individual is much, much greater, in fact, much, much greater than the bare skin individual, which was really interesting. I actually kind of expected this, but I wasn't expecting it to be quite so dramatic. And so we were really interested in why this might be. So I'm going to go back to some of the figures that I showed you before. If you recall our model of the chemical uptake through the skin, there's these different resistances, if you recall, the convective boundary layer and the stratum corneum of the skin. When we put on clothing, we're actually changing this convective boundary layer. Basically, we're replacing it by clothing. Now, the clothing, if it now has equilibrated with the air around it, basically reduces the distance between the contaminated air next to the clothing and our skin. This gap is really important. And so the analogy here now is that the gap, as the gap gets smaller and smaller, this pipe actually gets bigger and bigger. And so the resistance to uptake goes way down, and so you have higher, basically, flow through this pipe to us. And that is why we see such a much greater uptake from the exposed clothing than from bare skin, or from clothing than from air to bare skin, I should say. So we actually studied a few other chemicals this way over the past few years for a number of different reasons. One was basically we'd like to verify that this is happening with other chemicals, not just the ones that we studied. We were also interested in some of the more technical details of how this happens. Nicotine's a really interesting compound because it's actually a little alkaline, and our skin is somewhat acidic. And so there's a sort of hypothesis that basically we had a sort of two hypotheses. That there's a possibility that nicotine would not make it through the skin readily from air because once it got there, it would ionize. And ionized chemicals don't readily move through the stratum corneum of the skin, so it's a really good barrier for ionized molecules. But we weren't sure if that was going to be the case. If you were trying to break an addiction, we do know that nicotine goes through the skin fairly readily, but what the manufacturers of these nicotine patches do is that they adjust the pH of those patches so that the nicotine stays as much as possible in a neutral form, non-ionized form, and goes through the skin. And we weren't sure that would happen if nicotine would go from air to skin and further. So what we did was we took clothing and allowed them to equilibrate with airborne nicotine in this little chamber basically made of a shelving unit and some plastic wrap. It actually worked quite well for this particular experiment, and that's one of my students, Azine Eftekari, in the middle there. You see on the right a participant. This is one of the organizers of the whole experiment. Gabriel Becco. He is wearing clothing that has been exposed to nicotine. And you also see the clothing in the back there. That's clothing. Actually, I think he's wearing clean clothing in this experiment. In the back are the clothing that we had pre-exposed, and we also had it hanging in the chamber that had an elevated concentration of nicotine, with the purpose of just making sure that they stayed at the concentration we wanted them to stay at. Yeah, that person's wearing laundry clothing. So that's actually a sort of a protective condition. You'll see that they're wearing gloves and socks in that case. Let me show you the results from this experiment for a single individual. So on the left-hand side of this figure is, or I should say on the y-axis here, is the excretion rate. So this is a little bit different way of looking at it. Instead of the cumulative amount that was excreted, we're looking at the rate. And I like looking at rate because it kind of gives you a dynamic feel for what's happening here. So the person who's wearing clean clothing in this case, the one this is clothing that's been laundered, you see that there is some uptake of metabolites. You see the parent compound nicotine, and these are two metabolites in the urine. When you look at them, it sort of goes over, you do see some uptake, and this is probably because nicotine does pass through clothing a little bit. Clothing is still quite protective, it's just not as protective as it was for dibutyl phthalate in the previous experiment. When the individual is wearing a shirt that has equilibrated in the chamber with nicotine, you see a much greater output of these metabolites. And the rate at which these metabolites go through make a lot of sense based on what we know about the transport through the skin and the rate of metabolism. And each individual, I'm not showing you all of them, each of them had the same response. We see that that the clothing is a source when it has been allowed to accumulate these chemicals from the environment. We can compare this nicotine uptake to that of bare skin subjects. This is a funny axis. I won't go into the details, but basically this is a way of comparing all the volunteers and human subjects in a way that makes them the same. Essentially, it normalizes by the person's surface area and the actual concentration they were exposed to, things like that. So as much as possible, it puts them all on the same basis. When you're looking at bare skin subjects, you see here a similar range from roughly 0.8 up to maybe 1.7, maybe at most a factor of 2 difference, but I'll talk about this one in a second. But for this one, and this one, they're within a fairly narrow range of uptake for bare skin. There are actually 2 of these individuals that showered after the experiment to see if that would have an effect, and maybe it does. There weren't enough experiments here to show for sure whether it was a significant difference. But what it did tell us is that during the experiment, a lot of nicotine passes from the skin oil on the surface of the skin into the stratum corneum, where it pretty quickly, where it can't get washed off readily. So now let's compare these guys with these participants that were bare skin with the ones that are clothed. These are the clothed subjects. You see, this was a subject that was wearing fresh laundered clothing, and this is that same subject wearing clothing that had been exposed to nicotine in the air. And the same thing here. These are basically the clothing and the air for all these experiments are all the same chemical activity. And you see that basically it looks just like you're a bare skin subject, or you actually have a much higher value. And this is a fairly meaningful, again, it's only 2 people, so we can only say that it's at least as much or maybe a little more from clothing in this experiment. The amount that comes from actual air is quite small. Most of the uptake is coming from clothing, the green bar here. We also looked at a compound called oxybenzone. So oxybenzone is what's called a UV inhibitor. It's a compound that you commonly find in sunscreens and actually makeup. It's also put into plastics and coatings to protect them from UV damage, like you can imagine a coating on a piece of furniture. This compound, which we also call BP3, is metabolized to a compound called BP1. And that BP1 is the one that is probably of more concern. There are more health concerns associated with that compound. We're all exposed to this all year round, but there are seasonal differences. As you can imagine, you would see more during the summer than in winter because people use sunscreen more in the summer than in winter, and that makes sense. And you also see it more in women who wear makeup and then ones who don't. So there's definitely a relationship with the kind of materials that I should say, consumer products people use. What's interesting, though, is that it does show up in winter, and it also shows up in children in winter in a way that is surprising in the sense that they don't have any direct exposure to any products that have these chemicals in them. They're not wearing sunscreen, they're not wearing makeup. And so there was a hypothesis that came from some folks at the Royal Hospital in Copenhagen that maybe this was coming from clothing. And so we actually are interested in whether this mechanism would actually also play out with this compound. So we did a very, very similar experiment. I won't show you all the details here, but I'll just show you the result in which we took individuals and had them wear clothing that had been exposed to the parent compound, BP3, in air. It was just airborne uptake. And you see here the excretion rate of BP3, the parent compound, and its metabolite over time. And you see when they wear it and after they wear it, you see a rise in the excretion rate in urine, and then it drops down. There are differences amongst the participants, which in this case probably has a lot to do with differences in their skin. And I won't go into that, but that's a important consideration when we're thinking about exposure to the whole population. And speaking of the whole population, that can be a bit challenging as well, thinking about how do we use this information to make predictions. How much does clothing influence our overall exposure to pollutants, either indoors or outdoors, or anything that could be associated with clothing? And we have to ask the question, is this dermal uptake or transdermal uptake from indoor air important? And is the clothing route important? How does it compare to other routes of exposure? We know that we are taking in a lot of chemicals of different sorts in different ways. We eat things. There's dust in our houses that we can ingest or breathe in. We drink water that can be contaminated. There's a lot of other, and you can think of there's occupational exposures that can occur. So the way we can do this is we can take and try to develop mathematical models of the uptake by our clothing and bare skin pathways from air, indoor air, and compare it to the total body burden in the population. And I'll just go through really quickly here the sort of conceptual model. So the conceptual predictive models really just are, you have air, it's in contact with clothing, there's absorption that occurs, molecules can diffuse into and through clothing, there's a gap between clothing and skin, and those molecules can transport between skin and clothing and through skin, and then ultimately into blood and tissues, and then there's metabolism. You can build models that are layered in this way that ultimately provide a overall mechanism, a mechanistic way of predicting the uptake through skin. There are a lot of uncertainties. I'll mention a couple of them as we go along, but I'll give you an example of an output. So this is one of our experiments, the excretion rate of dibutyl phthalate from a person wearing clothing that had been previously allowed to accumulate this compound from air in a room. And what you see is that the experimental results here that are in red are very similar to what we expected, or at least what we predicted using this mathematical model that actually includes all of these parameters from the air of the room to the blood and the metabolism. It's not perfect, but it's actually pretty good. And we did this for a number of compounds in a number of situations, and came to the conclusion that it's close enough that we feel confident we can make some predictions about the population exposure. I realize now that one of the slides I wanted to put in here is not here, but I can just tell you the result. When we compare our model to what we know about the body burden from making measurements in the urine of a cross-section of the population of the U.S., we estimate that for a compound like this, like dibutyl phthalate, clothing and indoor air dermal uptake is probably accounting for maybe 10 to 40% of your uptake of that compound. Now, it may not be true today. This was based on some data from a few years ago because of the over time, the body burden in the population changes as chemicals get phased out and phased in. But at the time, we felt that for a few years ago, when we have that data for the body burden, also for conditions in homes, it appeared to be about 10 to 40%. So it's meaningful. But it turns out that's not true of all chemicals. And so some chemicals that we've studied probably do not have a major source. That clothing is probably not the major source, even though you find these chemicals indoors. So what are the considerations here, and where do some of the uncertainties come in when we're trying to make these predictions? Well, a big one is the history of clothing. Where are you hanging your clothing? Where is it accumulating chemicals from its environment? How long does it spend between washings? How well does washing remove the chemicals? For many chemicals, we do know that washing is not very good at removing these chemicals. Many of these larger molecular weight phthalates are only partially removed by laundering. And also, the process of laundering can actually transfer chemicals from the laundry machine to the clothing, or you may pick up some chemicals from the detergents and things like that. I'm not saying that any of these are particularly bad. I'm just saying that there's a transfer of chemicals going on all the time in any environment. Another important consideration is how these chemicals interact interact with the fibers themselves. In a sense, what's the capacity? And so it is true that different fibers have different capacities. So I know I often get that question, what about different fibers? What about polyester versus cotton? Well, yes, it does matter. In fact, it can be, the difference between polyester and cotton can be a factor of 10 difference in capacity, but it really depends on the chemical. This idea of the coverage is important. The clothing coverage, of course, the amount covered that makes sense, but this gap between clothing and skin is very important, and it's one of those areas for which we don't have the greatest measurements, and that's somewhere where we do need to have better information. But you can imagine if you have loose-fitting clothing, like this colleague of ours who is wearing, usually wears quite loose-fitting clothing, you would have less dermal uptake from the clothing. Whereas you have tight-fitting clothing, you have more dermal uptake. And the difference between loose and tight actually doesn't seem like a whole lot, but it turns out that it does matter quite a bit in the mathematical models. Tight-fitting clothing can be quite influential. To sort of take this a little bit broader, we also know that if you have chemicals that have accumulated in clothing, then if they are volatile enough, they will volatilize into the air around you. And because you have this what's called a convective plume of air around your body because you're a little warmer than the air around you, these chemicals are basically drawn up into your breathing zone. And so the concentration of those chemicals, let's say dry cleaning solvents, is a little higher in your breathing zone than in the rest of the room. So there's a little bit extra exposure you get by wearing them. You can also get exposure to particles that have accumulated on your clothing. And there's been some interesting work that's come out of Berkeley associated with that. This is a really important and interesting area that I think we should be putting a lot more effort into, is looking at the possible uptake by mouthing. So children at certain age tend to put everything in their mouths, and you can see this is my grandniece here putting a toy in her mouth, but the other thing in her mouth, or other thing in her left hand there, this is something that she's had since she was born, and that thing goes into her mouth 100 times a day, or it used to. And it turns out that children put fabrics, specifically fabrics, in their mouths a lot every day. And if you consider that, you consider how much can be extracted by saliva, you can actually get quite a bit of chemical transfer. Let me give you one example. So how much would a toddler take in by mouthing if they were living in a former meth lab? Well, this is work we did a few years ago where we were studying building materials and how they accumulate methamphetamine. And a shout out to Rich Corsi and Dustin Popendick, if they're out there, because they were colleagues of mine on this project. We took clothing, exposed it to methamphetamine in air, and looked at how much would accumulate. I mentioned this earlier in the presentation. We accumulated, essentially, if there's 1 part per billion methamphetamine in air, which, by the way, is not unreasonable in a former meth lab, even if it's been cleaned up, although there's not very many measurements out there. you would see about 6 milligrams of methamphetamine actually accumulate in that t-shirt over time. It might take a week or so, but it would get there. If then a child starts putting this in their mouth and they mouth these textiles at the rate that typical children typically do, then they would take in about 1 milligram per day of meth by that route. Interestingly, that value is very similar to the prescription level. So I was surprised to learn as we were doing this that methamphetamine has historically been used historically prescribed for ADHD. It's an amphetamine, and I guess most ADHD medicines are in that class. And so it would make sense that methamphetamine would be used. I think it's not used as much for that, but it tells us that this pathway can provide you with a meaningful dose, essentially, of something like methamphetamine. And this is true generally of or I shouldn't say most, but true of many chemicals that can accumulate. Not all of them are particularly saliva soluble, so we have to keep that in mind. Turns out that methamphetamine is fairly water soluble. So what do we do about reducing exposure from clothing? Well, it's tricky. One thing is, this gets back to this question of what can consumers do individually to reduce their exposure to chemicals in their environment? The thing is, we can't do a whole lot. There's a few things we can do, but on the whole, changing behavior is difficult, can be expensive, and it's also sometimes impractical. And what we really should be doing is what we have been doing, is identifying the chemicals that are problematic, identify exposure pathways, and work with manufacturers to reduce that exposure pathway by either removing these chemicals from the manufactured items or changing the way that these items are manufactured in a way that we just take less in. It turns out that if you take clothing for many of the chemicals I've talked about and just instead of hanging them indoors, you hung them outside. Let's say you lived in a very dry, nice place like Phoenix. You might be able to do this, and this would actually keep the chemical load quite low in your clothing because the outdoor concentration of chemicals is quite low, of the chemicals that we're interested in. But as I said, it's probably impractical for most people, and so really there's not a whole lot. I don't make strong recommendations here because I do get asked that a lot. So now we know a little bit, excuse me, about clothing and clothing-mediated uptake of indoor air pollutants. What about other textiles? I realize that I've only got about 8 or 9 minutes left, so I'll probably move through this fairly quickly. But what I'd like to say is that there are, of course, other kinds of materials indoors, other textiles out there that we should be concerned about. There is evidence that dermal contact with furniture fabrics can be a source of flame retardant uptake in our bodies. We do know that there's not that much difference between clothing and bedding, right? So the bedding basically acts like clothing when you're asleep. So anything that's in bedding is also going to be in you. There's also evidence that the kind of that when you're sleeping, your exposure to the environment of that sleep zone is much, much greater. And so if there are emitted chemicals, compounds, pollutants from mattresses, from other materials during sleeping, you'll get a much higher inhalation dose. I wanted to say something about indoor surfaces and how important they are in our exposure broadly. Then I'll talk about back to textiles. It turns out, and I won't go into a lot of detail here, but there's a lot of transport to and from surfaces in buildings. And the rate at which this happens actually is very close to the rate at which things move from air to you. It's in this area, this mass transfer coefficient of 5 meters per hour is kind of a typical number. So you can actually take the surface area of a building and get an air cleaning potential from that by multiplying the surface area of the building by this mass transfer coefficient. And if you get the surface area as the surface area to volume ratio, I should say, you get ultimately a result of 10 cubic meters per hour per cubic meter. 10 cubic meters per hour, but ultimately per cubic meter of volume. This is telling us that the air exchange rate, effective air cleaning potential or air exchange rate in a sense, is very, very large if you were using surfaces for cleaning. However, that means they're also really, really good at recharging the air around with chemicals if they have already become saturated. Let me just go through a couple of examples here. So a few years ago, Mike Van Looy and Bill Nazaroff and others at Berkeley were studying nicotine in buildings and what happens to nicotine in buildings when you release it. And what they found is they were doing these periodic injections of nicotine into a stainless steel chamber that had some carpet in it, that the nicotine concentration would go up and then it would immediately drop back down again. And this was over many orders of magnitude. And this is because the carpet was just absorbing so much nicotine, it was just like this huge sponge for nicotine. Since then, there's been quite a bit of research studying chemical absorption into textiles in general, and I've shown you some of that information for chemicals and clothing. So this is just to reiterate that textiles just have a huge capacity to adsorb or absorb organic compounds. We can think of this in a couple of ways. One, what's the amount of a chemical in carpet, for example, relative to what's in the air around it in a home? And this is a chart I made for this purpose. If we just take one compound, let's take nicotine, there's roughly 10,000 times more mass in carpet than there is in the air around it. So this is a huge capacity. We can also look at it in terms of time. It takes about 100 days for nicotine to basically for nicotine to essentially equilibrate with the air around it. It's got so much capacity that if you want to change the amount of nicotine in carpet by opening windows and such, it's going to take a very long time for that to occur. That's basically what this chart is saying, this characteristic time analysis. So let's look at this in a dynamic way. So this is a simple simulation showing what happens when you have, let's say, some old carpet. Inner house is equilibrated with the house. You open a window, and let's say we look at one chemical, toluene. This is the relative concentration in air. So you go along, the window's closed. Then around 10 in the morning, you open up the window. The concentration of toluene will drop a fair bit because you have more ventilation. And the toluene would go down even faster if it weren't being released by the carpet into the air. But eventually it comes down, and then it goes back up when you close the window. Okay, this sort of dynamic here is associated with the fact that there's some capacity of the carpet to release toluene into the air over time. But toluene is not a really good example here because it's so volatile. Let's take a look at dibutyl phthalate. When you open a window, you actually reduce the concentration a little bit, but not much. And that's because it has this capacity to be released. There's so much in the carpet that essentially it gets released very rapidly from the carpet because there's so much surface area and has so much in it. And opening the window just doesn't do much for compounds that are in this class called semi-volatile organic compounds. If you also consider the other surfaces indoors, and actually other furnishings like couches and, let's say, just painted walls, things like that, it turns out that opening window does almost nothing. So this is something just to keep in mind, that because of all the surface area indoors, there are chemicals that opening windows and increasing ventilation doesn't do much about. It just doesn't change the concentration very much indoors. So here's one actual example from an experiment that was done in Germany, again, the Fraunhofer Institute, WKI. They basically allowed carpet to equilibrate with a bunch of different chemicals here. These are all different phthalates or similar compounds. And then they turned on a really massive air cleaner in the room, and the concentration of these chemicals dropped down when they did that, but not as much as you would expect from having such a big air cleaner running. Then they turned it off, and almost immediately the concentration of these chemicals rebounded, and it really didn't have any long-term effect having this air cleaner on. So what that's telling you is that the materials, textiles, can basically maintain a fairly high level or maintain the status quo in a sense. What is it's such a big reservoir. But if it's such a big reservoir, it actually has the capacity potentially to reduce our exposure. So let's say we have really clean carpet that goes into a building. That means that it's at low chemical activity. And this is a little simulation I did where if you put carpet into a building and the carpet is quite clean, okay, it's not emitting anything, it will just absorb what's there as much as it can of certain chemicals, okay, not all. And what it would do is it would reduce the relative concentration relative to not having it there by quite a bit, by 75%. And over time, its capacity would get used up. We're talking about years here. It takes a long time. And so your overall exposure during this period would be lower. If you replaced or cleaned the carpet every 5 years, you'd even have a better outcome. Now, I realize that there are some sustainability problems with this, and it's not something that you would necessarily would do, but it gives you an idea of the capacity of textiles to essentially alter, influence, and mediate our exposure to contaminants in indoor environments. So I want to conclude now just to say basically what I've already said. Clothing and other textiles can hold on to a lot of chemicals and be really, really big reservoirs. So that means that they can be very protective, but they can also be a big source of exposure. I think the intimate contact that we have with clothing is very meaningful and important, and it means that there, at least for certain kinds of chemicals that where dermal uptake and mouthing is important, then we really ought to be looking more at clothing and what's in our clothing. So we can clean textiles to try to protect us from exposure because we use textiles, they're part of our lives. If they're clean to begin with, and then in a sense they are protective, there may be a meaningful environmental or societal cost of trying to do this. And so I'm not necessarily recommending that. There's a lot of people to thank for the work that I've showed here, and I'm probably missing a few. There was a lot of institutions involved. So shout out to all the folks out there. And I think I'll just go right to the slide I was told to go to, which is the questions and answers slide. Thank you.
Thank you, Glenn. We've received quite a few questions, and they're still coming in. So for those of you that haven't submitted questions yet, now is the time. Our first question, our attendee asks, could you discuss some of the thoughts behind hanging the clothes inside out during the exposure testing, as was seen in the pictures?
Oh, sure. Yeah, that was intentional. We wanted to make sure that at least the inside of the clothing had equilibrated with the room. And I mean, it's an important question. If we had hung that clothing, I guess, right side out, you'd say, right side out, it is possible that the inside of the clothing would not have risen to as high a concentration during that time period. However, we've tested that clothing since, we actually were testing it at the time and since then in the lab, and determined that it wouldn't have mattered, that the inner and outer layers of the clothing would have gotten very close to equilibrium over that time period. It wouldn't have been exactly the same, but pretty close. So we probably, if we had done it the other way around, we might have seen a slight lowering of the excretion rate or total excreted amount in that experiment, but it would have been probably with our, I don't know, but it probably would have been within the noise. So hopefully that answers your question.
Thank you. Our next attendee asks, what products did you use to launder the clothes?
So the good question. We use as much, well, for all the experiments we've done, we've tried to use a fragrance-free laundry detergent. Some of the work we did was in Denmark, so I don't recall what the laundry detergent specifically was. It was a fragrance-free, simple detergent, and I don't know exactly what it was comprised of. In the U.S., I'd have to guess it was like fragrance-free Tide or something, just a normal laundry detergent. We weren't trying to avoid particularly what was in the detergent at the time, but frankly, we didn't know what was in the detergents. But more important in that process was after we did the laundering and before we used it, we actually hung the clothing outside for several weeks to get them to equilibrate with outdoor air, which is much cleaner.
Thank you. Our next question. I tend to wear clothes twice before washing them. This makes me feel that I'm being more energy efficient. Do you have a sense on how long it will take for our clean clothes to become exposed clothes?
Yeah, That's a great question. We've actually studied this quite a bit, and it really depends on which chemical we're talking about. For some chemicals clothing can have a different capacity for different chemicals and for different fibers. The higher the capacity, the longer it takes for it to become basically contaminated. But it's a continuum process. So I would say that for most of the chemicals of interest for dermal uptake, in this case, it's going to be on the order of a week to a few weeks before kind of starting to reach capacity in a sense. So it's in that time frame.
Thank you. Our next question. On another note, since clothes act as a protection shield and as a source, would you expect our hair, head, and body to act the same way?
Maybe. I guess it depends on how hirsute you are. The hair on your head probably does act that way. If you're washing your hair, it probably does act as something of a real barrier, just like your skin, the hair on your head or a beard. The amount of hair on the rest of your body, I don't know. My guess is less so, just because the density is so much lower than clothing. But that's not a question we've studied, so I don't know.
Thank you. Next question. Curious if different laundering techniques, including soap and temperature, impact the effectiveness of clothing as a protective barrier.
Yeah, we do know a little bit, but not a lot. So what seems to be more important is which chemical we're talking about. So for larger molecular weight phthalate plasticizers, laundering is not very effective, even using detergents. For smaller molecular weight contaminants and ones that are more water soluble, laundering is much more effective. It kind of makes sense. In terms of whether one large detergent versus another or one temperature versus another, I actually don't recall. There have been some temperature studies, and I don't recall the outcomes of those, and we'd have to look back at that. But I don't think they're really strong result. Actually, the differences between the typical temperatures that we use for laundering wasn't huge, but I don't recall exactly. It'll have some effect. But also consider when you're drying clothing. If you're drying clothing in the house in a dryer, an actual electric dryer, the air that goes through your dryer is actually coming from your house. And so if you think about it, let's say you dry the clothing at some low temperature, gentle cycle, you're essentially rapidly re-equilibrating the clothing with the room air in a sense. So it's just like you're hanging it in the room. But those are studies we actually haven't done. That's a more of a hypothetical situation. But it's probable that that's what's going on.
Thank you. So I'm seeing a lot of questions like this question that we just received, so I'll just ask and hope that it covers everyone else's. But cotton is grown with heavy use of pesticides. How much of a problem is dermal uptake of pesticides through wearing these clothes?
I don't think I can answer that question since we haven't studied it. I don't know how much of the agricultural pesticides go from the growing to the manufacturing. Most fabrics, or at least natural fibers, go through a lot of processes before becoming a fiber. And I'd be pretty surprised if much of those pesticides actually make it through the washing and processing of those. But it's possible. I just don't know. Sorry, I can't answer that one.
Not a problem. For our next question, can frequently ironing, refreshing of clothes drastically reduce exposure to once absorbed contaminants? Higher temperatures can be involved compared to laundry washing.
Yeah, that's a great question, and I don't know the answer to that. Heating with an iron certainly will drive these chemicals off materials. If you take them and heat them up somewhat, you'll see an increase basically in their emission rates from these materials. However, there's a limit, and we do know that it really depends on the fiber in this case. But for cotton, we know that these compounds actually don't just stick to the outside of the fibers, they also dissolve to some degree into the fibers. And heating it up, while it's going to help release what's on the outside of the fibers, what's inside is much slower to sort of release. So you probably do remove some of those chemicals that way, but we haven't actually done that study. It's an interesting question.
Thank you. Next question. Do these findings suggest that non-carpeted hard surface flooring is better for indoor air quality?
Yeah, that's it's a big question, and one that's been discussed in our field for a long time. There's a lot of factors involved, and there's no simple answer here. The thing about carpets, if you put in new carpets, it's a source of certain volatile organic compounds, some of which in the past were things we didn't want to be breathing in. And like styrene was an example. The emissions from carpet, when they're new, are better than they used to be. There's less styrene emissions, but there's still some volatiles that maybe you don't want to breathe in. It really depends on what condition it's in. If it's clean, and by clean I mean that it's free of the chemicals that we care about, then it's going to be a net benefit. It's going to be a sink. It's going to tend to reduce your exposure. But over time, it becomes basically it's this big reservoir. It accumulates chemicals from its environment. Not only that, it accumulates dust. It accumulates the debris that you bring in from outdoors. And so buried in an older carpet is quite a bit of not such great stuff. And also all the stuff you bring in can be a food for microorganisms. So I guess the question is, what is the history of that carpet? Whether that tells you whether it's a net benefit or a net negative, I guess. And if you were to ask, what do I have in my house? I go with what I like. So again, it's one of those things where it's a complicated problem. There's no specific recommendation I can make that benefits everybody all the time. In my house, I actually have some small carpets, like throw rugs, I guess, that are there just because I like them, but on the rest of the house is just hardwood.
Thank you. Next question is sort of a follow-up to this one. Were any of the carpets cleaned to see if their ability to remove chemicals from the air rebound?
Well, in the studies that the answer is not that I know of. So some of the studies that I was describing are by other researchers. I've studied carpet as well, and I don't think the ones in Berkeley were cleaned to see if there was a rebound effect. And so I really think that's an important question. It would be really, we haven't done those studies, especially we haven't done those studies in the field where we've taken carpet sort of at a known chemical activity, either zero or something else, and put it into a building and looked at how it affected the the concentrations of those chemicals in a real building. That we can predict what would happen there, but we don't know exactly until we do the experiment. So I think that'd be a great thing to have somebody fund.
Thank you. Does carpet cleaning introduce new exposure agents? For example, clothing detergent?
Yeah, the answer is I don't know. I did have a discussion with folks that do carpet cleaning and that do that are from sort of that world a few months ago, and they do use some detergents to take care of some of the spots, but mostly the ones that I talked to were trying to stick as much as possible to just hot water and steam for most of the cleaning. But the answer is I don't know what everybody's using, so I don't know whether or not there's a significant post-cleaning exposure. In general, I'm not real fond of any kind of cleaners that have fragrances in them. Partly is that I just don't care for it, but a lot of these fragrances are quite persistent. And it's just like anything else. Why put something into our homes if we don't know if there's a true net benefit? Just because it smells good doesn't mean it's good. It doesn't mean it's bad. I'm not saying that fragrances are necessarily bad, but for some people, it's not they don't like it. Let's just put it that way. I'm not going to say anything, talk about sensitivities or anything like that, because I'm not qualified to. But generally speaking, I think you should just limit what goes into your home as much as possible.
Thank you. Our next question. I recall the clothes used were 100% cotton. Are there any future plans to see other kinds of textiles, such as polyester, wool, silk? And if so, how would these different types of textiles perform in your experiments?
Yeah, we actually did do some blend. I think in the nicotine experiments, those were blends, and they actually had less capacity for nicotine than the cotton. The blend had a less of a capacity for nicotine than the cotton did. And so we actually, I think, saw that in our experiments, that we saw a little bit of transport through the clothing. And so it was less of, and I think I don't remember the exact blend composition. It was something like, frankly, I just don't remember. But it is true that the composition is important. And so for many of the chemicals that we've talked about, the phthalates, the cotton is a really good absorber, and it's going to be a really good protector in that sense, but also can hold on to a lot. Polyester, nylon, not very good. They're quite a bit less capacity on a pound-for-pound basis. But interestingly, and this is still in this, we don't have enough data to really firm this up, but spandex seems to have a really, really big capacity for these chemicals. And so just adding a little bit of spandex to, say, cotton like jeans really, really increased the capacity of the jeans for these plasticizers. But again, it's just a little bit of data we have there, so I can't confirm that absolutely. But yes, it really does matter. So we've moved, worldwide, we consume more synthetic fabrics, synthetic fibers in our clothing than natural fibers now. We've moved from across that line. And so in general, tech clothing is on the rise because it does so many great things. So it's really, we can do so much with it, and we like wearing tech clothing. And so I would expect, if you looked at like the long term, that a move away from natural fibers to tech fibers would see a change in our exposure pathways a little bit. I don't know which direction for sure it's going to go, because some chemicals interact more strongly with synthetic fibers, some interact more strongly with natural fibers. It really just depends on the actual structure of the molecule and the composition, the chemical structure of the fibers, whether natural or synthetic.
Thank you. For our next question, what about layers of clothing? In wintertime, people often wear 2 to 3 layers on some parts of the body. Which layers are most influential for what gets through to the skin?
Yeah. So the outer layer is going to be the one that's most influential while you're wearing it, for protecting you from what's in the air around you. The one that's closest to your skin is going to be the one that actually is controlling your exposure. And in the sense that the one that's closer to your skin is the one that, so if you take clothing from let's say just as an example, let's say it's winter, as your example, and you're wearing long johns, long underwear, and you take that out of the drawer, you put on the long underwear, and then you put on your clothing over that and your jacket, and you go outside, and then you go hiking somewhere, you're out all day, you come back later in the evening, and you take it off. The thing is, that whole time that you were out hiking, you're outside, you weren't at your home, the clothing you were wearing, the long johns were the most important exposure source for dermal exposure. And the thing is, that clothing effectively looks like the drawer it came out of. So the entire time you were out all day, you were wearing that underwear drawer, if you think of it that way. And so if you were able to look in some sort of fine-tuned way at your dermal uptake of chemicals, it would look like you were at home, or at least it would look like you were somehow sitting in this drawer. I don't know. That's not a very good example in that case, but the idea is that the clothing that's closest to your skin is what's really what's influencing your direct exposure, but what's protecting you from what's around you is the one on the outside.
Thank you. All right, so our next question. how receptive have clothing manufacturers been to these scientific results?
Well, frankly, I haven't really talked to many of them about it. I'm interested in exposure pathways, not necessarily in changing anything about the clothing industry or manufacturing processes necessarily. I've spoken to a few people about these things, but it's sort of in this realm of that's an interesting result. I don't think that really affects what we're going to do. However, I've said that I have talked to some folks, some small clothing manufacturers, about whether or not it would be possible to incorporate these findings into clothing. How do I put this? Into providing clothing with an added benefit. Of course, every company is going to look for a way to set themselves apart. And if they can say, oh, this clothing has been cleaned in this way, and we've shown with human subject experiments that it helps protect you from dermal uptake of all of these chemicals from the environment while you're wearing it, and blah blah blah. Okay, so you could theoretically do that, and that would be a very niche market. And I know this. I would say that it's probably one of those things that probably only more wealthy people are going to be interested in, doing, or basically purchasing this kind of clothing or a clothing service like that. But in terms of like the larger picture, I don't think this is going to change that, because this is just part of our environment. The clothing, I don't think we should be changing the way we make clothing necessarily. It's just, I just want to understand these pathways so that we can identify how these chemicals are getting in our bodies, and then say, okay, if there's a pathway that's a problem, we need to go to the easiest way to deal with that problem. It is most likely removing that chemical from the consumer products in the first place, so it never ends up in our house at all. And most of these chemicals I'm talking about are not coming in with the clothing. That's not the issue. There are some problems with some clothing. There are dyes that can be problems for people who have sensitive skin. I won't go into those details. Like I said, I wasn't going to talk about that today. But most of the chemicals I'm talking about are ones that are coming in other products. They're coming they're in the flooring, they're in the consumer products, they're in your couch cushions. That's where we should actually start. The clothing just happens to be a transport vehicle for these chemicals to get into us. Now, that said, we can't fix this problem immediately, because all of our buildings, all of our homes basically are contaminated with lots and lots of chemicals that come from manufactured products. I say contaminated, it doesn't necessarily mean that these are bad, they're just there. And contaminated is probably a bad word for that. However, if we said, well, who are the most vulnerable in our population for endocrine disrupting chemicals, say broadly speaking, things like certain kinds of plasticizers, certain kinds of flame retardants, things like that, maybe there's an opportunity in that population, children, pregnant mothers, that sort of population, where we might be able to have an opportunity where we can use clothing or something similar during these time periods to help reduce the body burden in homes. But I think that's kind of a stopgap, and we really ought to be going at the original source of these chemicals as our most effective way to reduce overall population exposure. Sorry, I got off on a tangent there.
That's all right, thank you. So I'm seeing a lot of questions on biological contaminants, like mold, for example. So could you say something about, or talk a little bit to mold, biological contaminants, and how that relates to the results that you saw in your study?
Well, we have not been looking at biological contaminants, and that's a good question. It's a big question. If clothing can be also a carrier of biological materials like pathogens, like dead skin cells, fungal spores that can move around a building, and so because they can accumulate in clothing, they can also come off clothing and into your breathing zone. So in a sense, that it also is a transport. Clothing can transport these things. Whether that transport to, say, your skin is important, I don't know. that's outside my expertise. I would say I don't really want to. I guess because most of these are fairly far out. The idea of how influential clothing is in your exposure to biological materials is something that that we wrote a review paper on recently. But the thing is, the data is relatively sparse, and so the places where there's more data is in the hospital setting, in the medical setting, and asking how well is clothing being cleaned of pathogens, and how do these things like the skin cells of the medical personnel moving from themselves to their clothing and then into the environment around them, how can that influence exposure to what we think of as a fairly clean setting? That is a complicated and big area, and clothing is an important part of it. But beyond what I've already said, I don't really want to step too far into that, because I think there are some other people out there who can speak to that better than I can.
Thank you. And for our final question, since we're nearing the end of our time, this attendee asks, will air purifiers reduce clothing or carpet chemical exposure with closed windows?
So I don't think air cleaners are, they're generally not going to be particularly effective in this case. And that's partly because, well, you can imagine if you're wearing clothing, if the major sort of exposure pathway is through your skin, having an air cleaner in the room isn't going to do a whole lot of good. Air cleaners can be useful in certain cases, and there's a lot of caveats, and I won't go. I am already seeing all the possible ways this question can go, but where air cleaners really, I think, are effective in buildings is in particle control, especially let's say there's all these fires in the West. Having an air cleaner in the house can reduce the air concentrations of smoke particles. So that's, I think, a reasonable way to apply things. It's also, air cleaners are quite good, can be good air cleaners at reducing the concentrations of allergens in the air, especially perhaps if you use it in your bedroom at night. So I think there's a place for that. For chemical contaminants in air, in theory, they can do this, but there's not much on the market I think I would recommend for this, because you have to have a really, really, really big airflow through these air cleaners in order to have a meaningful reduction in the concentration of these chemicals. And that airflow is more than you can get normally than even just opening the windows of the house. So the best way to have clean air, if you live in an area that has good outdoor air quality, is just open the windows. But that's not always possible in a really hot, muggy summer or a really cold winter. Do air cleaners help? They might help a little bit, but frankly, for removing those kinds of chemicals from air, I would say it's probably not particularly effective, especially for the semi-volatile organic compounds. It's just not. I'm sure we could find it. If you put a bunch of air cleaners in a house, you could probably do something meaningful. But then that's pretty expensive. It's a large energy input, and it's just not, I think, sustainable across the population for people to be doing that.
Thank you. I'll now turn it back over to Allison Clune to close the webinar.
Thanks, Christina, and thank you, Glenn, so much for joining us today. This has been a fascinating presentation, and I'm really glad that you could be here. Thank you also to everyone who joined us online for submitting your questions. For announcements of future IAQ Science webinars, please visit epa.gov/IAQ, and we thank you for joining us today. Bye-bye.
Thank you.